if you heard those words, you've got cancer, you've probably been through some treatments and you've probably had some side effects from those treatments. And we're going to talk about that today. There potentially is some good news coming out in the world of oncology when it comes to figuring out what treatments are going to work for you specifically so that you don't have to go through all of the side effects with treatments that don't work. So I'm looking forward to introducing you to Dennis Watson with Travera. So let me roll the intro and we'll dig in. If you heard the words, "you've got cancer," you understand the overwhelming flood of fear and uncertainty that follows that sentence. I get it. I was there too, diagnosed with stage 4 ovarian cancer with an 11% shot of survival. But 11 isn't 0, and here I am living my best encore. My name is Melanie Holscher, author of Becoming Ovary Jones, How to Fight Cancer Without Losing Your Mind, and the host of The Ovary Jones Show, where we talk about hope, lessons learned, and the blessings hidden in the storms of life. Tune in and share with a friend, because nobody should fight cancer alone. Dennis Watson, welcome to The Ovary Jones Show.
Thank you, Melanie. I'm happy to be here. Thank you for having me.
Well, as a survivor myself, I have gone through a lot of treatment and I have felt the side effects and they're not fun. So you're going to talk to us a little bit today about something possibly, hopefully, maybe that could be a little bit of hope in the world of oncology. So what's going on in your world? And you're with a company called Travera?
Yeah, I'm with an organization called Travera, and Travera was spun out of MIT, the Massachusetts Institute of Technology in the Boston area, and did a lot of its early work with Dana-Farber as well. But we have an incredibly cool, very new technology that's sort of disruptive in the cancer space. And we're helping to guide treatment in late stage disease in a very new way.
Wow. Okay, that's pretty exciting. Now, anybody that's gone through a cancer diagnosis, we would love to be able to know what the next step is going to be with some authority and really have some confidence that whatever treatment we're going to go through is going to work. But in reality, we kind of feel like guinea pigs. So you're trying to help us a little bit. What are you doing that is helpful potentially? This is all brand new, I get it. Potentially to give us a little bit of confidence that what we're going to subject our bodies to might actually work.
Yeah, so you set up the problem really nicely. The challenge in late stage disease um, becomes therapy selection at a certain point, and there are excellent guidelines that exist that are backed by years of data and research that oncologists follow when they're treating cancer patients. And at the newly diagnosed stage, early cancer diagnoses especially, the drug options are good. The guidance that's available in those guidelines that are driven by research and clinical studies, it's good. The problem is when you get into later stage disease, especially if you've been through a couple lines of treatment. You know, our test is a pan cancer test, kind of works across almost all carcinomas and a lot of the blood cancers as well. But given your personal experience with ovarian cancer, we'll focus on that a little bit for the context of this conversation, just to keep things simple. If you have recurrent ovarian cancer, let's say that you're not platinum sensitive, and you go into the NCCN guidelines or the ASCO guidelines or whatever the doctor's using to guide their therapy choices, you're faced with a large page of drugs. They're categorized by cytotoxic chemotherapies and targeted inhibitors and immunotherapies and hormonal therapies. But within each of those categories, the drugs are listed alphabetically. And there's something like 30 plus drugs on that list. And the same thing tends to be true across just about any tumor type you might have. And so it's not just a problem that's specific to ovarian. The challenge is that the guidance when you get to that stage is really minimal. It says, here's a lot of drugs that are indicated for use here. We're not completely sure which one should come next. And so you've been through the first two lines, you've been through that first line of therapy that everybody kind of gets, and the second line, the follow on, where maybe there's a couple of options to choose from. And you get to maybe that third line, and you're facing recurrent disease, and you're sure as heck not done. You're not
Mm.
done fighting. You're not done pushing forward, but you don't want to be a guinea pig. You don't want your doctor to make an educated guess of which of those 30 therapies they're going to select next, because it does become a game of cat and mouse, or a game of I'm a guinea pig. It's, we're going to try this, we're going to see how it works, we're going to see how you tolerate it, we're going to do some imaging, we're going to, and then we'll make a determination if we keep going or not. And that is emotionally exhausting. It's physically defeating. It's a really tough place to be. And so, where our assay comes in is it helps with that selection. It helps to say, here's a list of drugs. Let's figure out which of these drugs have the highest likelihood of working against your specific cancer, your really unique situation.
Absolutely. So that sounds absolutely fantastic, right? However, we've been trying to do this for a long time, right? Try to figure out which ones are going to work, and we haven't been able to crack that code yet. So and the ability to be able to test for treatments outside the body so that we as cancer patients don't have to go through all those adverse events and the mental thing of when a therapy doesn't work. It's brutal. But again, little skeptical because we've been down this road before. We've tried to crack this code before. What's different?
Absolutely. So, and I didn't clearly describe this yet, but what we're essentially doing is called an ex vivo drug test. And
Yes.
you hinted on this, but it's the idea that we take cells out of the body and we test drug response against those cells, um, kind of in an artificial environment. The history of that comes from antibiotic susceptibility tests, things called ASTs. And for many, many years in the infectious disease world, if you get an unusual infection and you go see your infectious disease doctor, they're going to take a biopsy of that infection, and they're going to culture it in a petri dish. They're going to try 15 different antibiotics against it, and they're going to give you what works. And it's wildly effective. It just makes a whole lot of logical sense. Yeah, let's figure out what drug kills these cells before we put it in my body, right?
Yes, absolutely.
So as you said, we've been trying to do this in cancer, and since the 1950s, there's 70 plus years of research behind this. There's a challenge though that has always existed, and the challenge is that when you take a cell out of the body, you remove one of those cancer cells, really any cell. Those cells on average only live for a couple of days, 2 or 3 days, before they just naturally die off. They don't have a life source anymore, right? They're not being fed and nourished and things. And so we have to do things to keep the cells alive. And that's historically what we've done. Most chemotherapies and cancer drugs take about a week to take effect. So if you want to see if a cancer drug can kill a cell, the cell's going to die in a couple days all by itself, and the drug takes a week to work. You have sort of this time conundrum that you're trying to solve. And so the way we've solved it for 70 years in these ex vivo drug tests, they've also been called chemosensitivity tests in the past, was to intervene with the cells. We pump them full of proteins and cytokines and all of these things to get them to grow in artificial environments. You want to basically create clones of the cells, for lack of a better word. Sometimes they put them into mice and grow artificial tumors in a mouse. Sometimes they grow things called organoids, which is sort of like a three-dimensional tumor that's built, kind of like in a petri dish, but instead of Everybody grew petri dishes in their high school biology class, right, with the bacteria. But these grow sort of three-dimensionally, and the idea is they maintain more of the microenvironment of the tumor and the way the cells interact with each other. The challenge with all of these things, and the reason that it hasn't really taken hold in cancer treatment in the 70 years that we've approached it, is that when you intervene with cancer cells to that extent, they change. Unlike bacteria that loves to be fed and just likes to spread, cancer is a mutant, right? I mean, that's by definition what it is. It's your own cells that have started to mutate and have gone down a different path, right? And we've all seen enough Teenage Mutant Ninja Turtles to know that mutants keep mutating, right? And so when you intervene with a mutant and pump it with these proteins and try to grow it into these organoids or put it in these mice and you do these different things, we see changes in the tumor. We see changes in the cell. It's not always significant, but it can be. And it's that shift in that cell in those cells, they call it a phenotypic drift or a genotypic drift. It's slight changes in the way those cells behave. What we end up doing then is testing drug against cells that don't necessarily represent what's actually in your body. This has been the issue. And so, in a very long-winded way, to answer your question, the way Travera has solved this is that for the first time in 70 years of this kind of research, we're actually testing drug response directly against the cells that are removed from the patient. We're not intervening with those cells in any other way.
That's
They get
absolutely
removed
incredible.
via a biopsy, or if a patient has like ascites or a pleural effusion in their chest, they get that fluid drain. That fluid often contains cancer cells. So those, you get a biopsy done, you get that fluid drain, it gets sent to the lab overnight, and then we're able to measure drug response against those cells in such a rapid way that we can do it before the cells die off, and we don't have to grow them. We don't have to pump them full of all this other media and stuff to keep them growing and keep them alive. So we're really getting, in our belief, a much truer view of drug response in these cells than we've ever been able to do before. And it's at the end of the day, it's all about timing. It's the fact that we can do the entire test in less than 2 days once we receive it, about 2.5 days from the time the biopsy is done to the time we're sending reports to your doctor that says, here's 20 drugs. These six worked really well. These three or four did, okay. And then the rest of the list did nothing. They were basically placebos against these cancer cells. And it helps to provide a much more specific guidance to your unique cancer, your unique cells about what drugs are working there and what drugs aren't.
Absolutely. So very personalized and not taking weeks and weeks to figure it out, a couple of days. So that is absolutely incredible. I can't even imagine being able to have that test, get the results, and have more confidence that whatever treatment I'm going to go on, yes, I might still have to deal with the adverse events and the side effects, but at least I have more confidence that it's actually going to work. And I'm a lot more inclined to, push through those side effects. That's pretty incredible.
Well, yeah, that's exactly the goal, and it's about increasing the likelihood that we see a response. What's I think really interesting about our technology is that it's actually not a biologically based solution. It's a physics based solution. And that's what's made it very different than the 70 years of research that's come before. I think it's important to kind of describe how the assay really works.
Absolutely.
And what's cool is it's all about cell mass. And so, when a cell dies, it changes its mass. And any cell that's going through cell death, it's shedding things, it's losing parts of the cell through that process. It's changing its weight. And on average, when a cell dies, it loses about half of its mass in death. And so what's been developed at MIT is a new measurement tool, and it's a MEMS chip. A MEMS chip is like a microchip, but instead of containing circuits, it contains mechanical movement. Is the simplest way this layman can describe it. And we've got little fluidics channels in this thing, and so we get a cancer cell specimen and we pump the cells through this little chip one at a time, and inside that chip is what's basically a diving board. It's a cantilever beam, and as cells pass over the end of that diving board, the frequency of that diving board changes. And so imagine jumping into a swimming pool and you jump off of a diving board and you get that vibration from the diving board. It turns out that the vibration of that diving board in a pretty basic law of physics is dramatically different based on how much the person that just jumped off of it weighs. And so, assuming, Melanie, that you and I don't weigh exactly the same amount, if we both jumped off the same diving board, we're going to get a different frequency of vibration in that. And so, with that vibration, with that sort of frequency change, we're able to recognize the change in mass of an individual cell that would be equivalent to it losing about 5 nanometers of diameter. Um, I know those are terms most people aren't deeply familiar with. So the best way that I can describe it is the wavelength of visible light, a single wave of light, is about 400 to 700 nanometers on average. So we can see the change of an individual cell that's 100 times smaller than an average white light wave. Um, that just gives some indication of how specific this is. So the team at MIT basically said, we know that when a cancer cell dies, it loses half of its mass on average, right? We know that when a cell is responding to an effective cancer therapy that's killing that cell, that process can take about a week. So the question was, if the cell is responding to a drug that's working, is it going to maybe change its mass by a tiny amount, maybe just 3 or 4% in the first day? And so they worked with Dana-Farber Cancer Institute on that, and they found out that the answer was kind of a resounding yes. It turns out it's almost a universal biomarker. It works across all mechanisms of actions of drugs for the most part. We've already validated well over 100 drugs on this platform, 39 different tumor types. It's incredibly broad in its application, and essentially what we do is we get cells, we separate out the cancer cells from all the other stuff that exists in the specimen. We put some of the cells with drugs and keep some of them as controls, and then we incubate it for 24 hours. And then at the end of that 24-hour period, we go back and we weigh all the cells in every single specimen. Some of them are drugged, some of them not. And if the drug cells are changing their mass at a different rate than the control cells, that's what tells us that this drug is beginning to work on these cells. We see these cells changing, and they're changing faster than the cells that haven't been drugged. That's what tells us that these drugs are doing something. And the early data correlating that to clinical response in actual patients is incredibly encouraging.
That's amazing. So can you look at, you were talking about, testing 20 drugs, 6 that are having some efficacy, 3 that aren't or whatever. Of those 6, can you tell which ones seem to be shedding or reducing that mass faster?
Sort of, yeah. So we actually score every drug on a scale of 0 to 100. 0 meaning that there was no mass change at all. There was, these cells weren't impacted compared to control. And 100 being these cells were highly sensitive. They changed a lot. Um, the the scale is less related to what the mass change specifically is, and more related to the statistical confidence that mass change is is related to the drug and not some other variable, if that makes sense. So, on that scale of 0 to 100, for a drug that's above a score of 50, those are the drugs that we categorize as sensitive. These are drugs that are likely to elicit a clinical response in a patient. And any drug with a score of 50 or above, we have about a 95% confidence interval that what we're seeing in these cells in response to this drug is because the drug is working in these cells and not something else. For the cells that score, or for the drugs that score below 50, it sort of is the opposite. It's saying that, yeah, in many cases, if they score a 0, it's because these cells didn't change off the baseline at all, or, compared to the control. They changed at the same rate that all the other cells changed. They really haven't done anything different or unique. If something falls somewhere in the middle, we use that 50 line as kind of the cutoff of, yeah, there was some change here, but we can't be really confident that it's related to the drug. Or we're pretty confident that it's related to the drug, or we're really confident that it's related to the drug. So it's all sort of positioned on this scale.
Okay.
What we commonly see in a 20 drug run, though, is that there's a few drugs, five, six, seven, it's different for every patient, of course, that are going to sit way up at the top, right at 100, that are really highly sensitive. And then we tend to get, 10 or 12 that are sitting on that bottom line at 0 that just did nothing. And then we'll have a few that are sitting somewhere in that middle range.
Okay. So you get your cells in 2 days or so, 2.5 days. My doctor gets a spit out of all of the drugs that are, 20 drugs, which ones were pretty effective. And then, of the top three or whatever, then my doctor can look and see, talk about side effects and things like that and make a decision that's going to be custom tailored to my cancer cells.
Yeah, that's exactly right. And something I should share. I said that the early data looks really good. You know, as you started the show today, it's new, right? There's a lot that we don't know, but there's a lot that we do know. And we have full outcomes data on roughly 60 patients so far. We've tested several hundred. But patients that have gotten the test, the doctor has given the patient one of the drugs that we tested, and then some months later, we find out, was there a clinical response? Did the tumors diminish? Did that pleural fluid go away? Things like that. And so far, our predictive accuracy based on that is 85%, meaning that when we say a drug is going to work and a patient gets that drug, we see a clinical response in those patients between 80 and 85% of the time. It's an incredibly strong indicator that this is a really good universal biomarker for patients in this situation. Um, and so the idea would be that you do this test and you come back with that list, as you said, that says, hey, here's 6 drugs that were highly sensitive. Based on the data we have today, we're going to say, for these 6 drugs, 85% of the time, they're going to work. And they're going to create some sort of a response in you. For these other 12 drugs that are sitting on the bottom, 80 to 85% of the time, they're not going to do anything in you if you take them. You're going to just continue to progress, or your disease is going to be unchanged as a result. What I think is helpful to do in the treatment paradigm that many patients and physicians are facing is, we're not testing a bunch of really random, unusual drugs. We're doing that ovarian cancer panel. We're running 20 drugs that are FDA indicated for use in late stage ovarian cancer. So we're not running a bunch of research drugs. We're not running, although we can customize a panel to a patient or a physician's liking. We're not doing things that are totally off base, that a doctor sees a report and says, I would never give this drug. This is crazy. This is really about taking a list of drugs the doctor's in many cases already thinking about and saying, well, look, you've got these 20 in your view. These five appear to be really sensitive at the cellular level. So let's narrow our focus to these five. Then the doctor can use a lot of their other insight about maybe genetic mutations that they know you have for some of the targeted drugs, or the side effect profile of one of these five or six drugs versus another one, to kind of make a deeper informed clinical decision on which of these five or six drugs might make a lot of sense to try next, and give a shot, because there's just a greater likelihood that it's going to work than the opposite. The other value on the other side of that is that, you know, maybe maybe the doctor isn't comfortable with one of those five or six drugs for whatever clinical reason that they have, which certainly is feasible. Um, but there's also 12 drugs, let's say, that we showed had no sensitivity. And often there's just as much value in ruling out things that you don't want to waste your time with as there is ruling in things that you do. And so there can be really a lot of value on both sides of that coin.
That's absolutely exciting, incredible. But let's talk about a couple things. First of all, how you get the cells. We go through so many tests and so much, and yet another test. How are you getting those cells out of our bodies?
No, that's a great question. And right now we're really early in our process, and so we're not really recommending patients go and get a biopsy specifically for the sake of our test. It happens, some patients want to do that, but in most cases, we're just taking advantage of biopsies or procedures that are happening for other clinically indicated reasons. Um, there are lots of different reasons, as you know from personal experience, that throughout your cancer journey, your doctor wants to get another core biopsy done, another needle biopsy for, to run these other tests or to do a new analysis or to see if the genetics have changed, things like that. And so what we try to do is just work with patients when there's a procedure already planned to say, hey, you're going in to get this biopsy. Your doctor's ordered it because they want to, do a genetic panel. While we're there, we're going to just have the interventional radiologist take a couple of extra cores to send to Travera, which doesn't create a lot of change for the patient and what's already happening. The second thing that's relatively common, well, in ovarian cancer specifically, in later stage ovarian cancer, many patients develop ascites, fluid in the abdomen. It becomes very uncomfortable. They get kind of distended, and it's just not a comfortable situation when you begin developing that fluid buildup. And so for patients that have that, they're often going in at regular intervals to get the fluid drained. And they're getting the fluid drained because it just provides comfort to get the fluid drained. In most cases, the clinics, the doctors aren't really using that fluid for anything. They're kind of just throwing it down the sink, for lack of a better description. And so it's actually a really valuable fluid for us, because it often it contains a lot of cancer cells. They're easy cancer cells for us to access through our process. And so somebody goes in to get an ascites drain, and we provide one of our test kits ahead of time in working with the patient and the doctor, and they just collect a small amount of that fluid into the vials that we provide, drop it into our shipping kit, and get it to FedEx. And so we get it the next day and run our process. So those are the two ways we most commonly get specimens.
Okay.
But in most cases, for the tissue biopsies, we're taking advantage of procedures that are already planned. And for the fluid drains, it becomes pretty easy because that's something that somebody's already having done. Sometimes patients, if they have a lot of fluid buildup, even get something called a Plerex catheter, where they get a permanent catheter placed, and they drain at home themselves, or they have home health do it. And in those cases, once we have the doctor's order and support in doing the test, the patients even in many cases have collected the fluid themselves, drop it into our kit, and send it to us that way.
Amazing. So, a couple things I think is really important to point out. First of all, the patient that you're looking for. Obviously, if you're out there and you're on a treatment and it's working, we don't want to disrupt anything that's working, obviously, right? So you're looking for patients that have gone through maybe first line, second line, they're advanced stages. What patient profile exactly is right for you as far as testing goes?
Yeah, no, that's a great question. And I think the concept of this test applies kind of anywhere. The reality is that the data is just too early for that. For newly diagnosed patients that are on treatments that are working and with clear indications for what drugs they should be trying, the picture is relatively clear and the guidance is good. And so we should not be afraid to follow our doctor's lead there, of course. And the challenge becomes when you get into that later stage environment where it does become more of a guessing game. And that's where we feel like right now, at the level of data that we have today, that's where we can provide the greatest level of guidance. The reality is even, if somebody wanted to do it earlier, a lot of physicians wouldn't be comfortable with that, understandably so. The
Right.
guidance that they're following has so much data and history behind it to go off that path, off what is a really clear path, with an assay that is in early in its development phase as ours is, just doesn't make a lot of good clinical sense. But for that late stage recurrent disease, you've already tried, maybe 2 lines or more of therapy before. Um, it will become quickly clear that what line three is and fourth line and on is more of a guessing game. And there just isn't as much information on where to make those decisions. And that's, I think, the most valuable place for us. And as you highlighted, somebody that's looking to make a change. If you're having a biopsy done because the doctor is looking to change your therapy, if what you've been on has stopped working, or you've been between therapies and you've had a new recurrence come on, and you're at a point that you're about to change what you're doing and initiate a new therapy, that's the best time to do this. Um, as I said earlier, cancer is a mutant and cancer changes, right? And it changes over time in your body, in response to different drugs. And cancer constantly sort of can evolve in many cases. And so doing our test today and identifying the sensitivity of certain drugs and then going on some other drug and waiting 6 months and letting that stop working and then coming back to our results that are now 6 months old and using them for a care decision at that point is not nearly as valuable as getting a sort of a newly run test, because the reality is that your cancer could have changed. And so we like to do the test as close to a treatment decision as possible so that it sort of is real time in helping you and your physician to make those decisions.
What do we do whenever we go and we talk to our oncologist and our oncologist says, ex vivo, that doesn't work. This is not going to work. And they're skeptical, just like I was. Uh, what do we say? What do we do?
No, that's a great question. And I always like to help provide guidance there. One of the things that I know you and I talked about before, Melanie, was, as I said earlier, this is a really early test, and we're not reimbursed yet. We are CLIA approved, and so I'll just give everybody some of these terms. This is a laboratory developed test. It's called an LDT. There are hundreds and hundreds of LDTs out there. The approval process for LDTs doesn't go through the FDA at this point in time. It goes through a laboratory approval process called CLIA. And so this is a CLIA approved test. It's approved for clinical use in the US, and it does require a physician's order. This isn't something a patient can order for themselves. So we've crossed that threshold of our data set. We've got that CLIA approval, and we're focused right now on continuing to build data and validation to move towards reimbursement from Medicare and private insurers so that this can become more available to more patients. And taking this directly to patients in whatever way we can, making it as available as possible as people can benefit from it is really what our mission is. And so, as we're continuing to try to build that data, the test isn't paid for by insurance today. What we are doing through groups like yours, and sounding boards like yours, I should say, is we're trying to make patients aware that we exist, and we're giving the test away. We're not charging anybody for it. And what we're asking for in return, because nobody does anything for free in this world, right, is just feedback on the experience, feedback on what happens in your case. And so we'll run the test at no cost. And what we're asking for in return is for you to let us know what happens. Did you end up going on one of the drugs we tested? Did it work? Did it not? And we work with you and your doctor through that whole process. And so rather than taking this directly to your doctor based on what you've seen here today, what I often recommend patients do is reach out to me. And the reason I suggest you reach out to me first is just because I can provide you with a little bit of support and guidance for when you have that conversation. Because a lot of doctors are going to say, yeah, chemosensitivity testing, ex vivo drug testing, we've been trying this for years. It doesn't work. It's not something that I do. We understand that hesitation, that skepticism. Often when I have an opportunity to talk with an oncologist directly and describe how unique and different our technology is compared to what's been done in the past, the response typically is, okay, that sort of makes sense. It's worth a try. And there's not a great deal of risk, right? We're working off of procedures that are already happening. We're running drugs the doctor's already considering. We're not making any, we're not here to make some crazy recommendation. We're not going to say, hey, you should give this ovarian cancer patient this lung cancer drug. That's not what we're trying to do. We're just trying to narrow that scope for the physician and allow them to make the most informed decision as possible. But the way that it gets brought to your doctor can really matter. And so what I often recommend is if a patient's interested in getting this test, I have them reach out to me directly. You can go to our website. We have a contact us button that actually comes directly to my personal email. Or you can just email me. I know we'll include my email address in the description with the video here. Um, but once somebody reaches out, I sort of just walk them through the process, and I'll even offer to give them a draft email that they can use with their doctor to kind of introduce this. And surprisingly, I find that most doctors with patients that are in this position tend to be pretty open to the possibility of what we're doing. In fact, we really launched our CLIA lab, launched this test outside of just the formal clinical trials that we're still doing, just last November, so a little less than a year ago. And in that period of time, I've interacted with well over 300 patients, and the vast majority of doctors that we've engaged with have absolutely been willing to participate. There have been a handful that have said, you know what, it's too early. I'm not comfortable. I'm not familiar with the technology, etc., which is understandable. But most of the physicians we've interacted with and worked with and patients we've connected to, their doctors have said, you know what, let's give it a try. Why not?
That's exciting. I think it's also really important to note that it is 2023. So I have people that find the Overy Jones show and they watch episodes from 2 years ago. So 2023, you are actually providing this test for free to collect data. And I think it's exciting for anybody to be able to be a part of research and the future, hopefully, of oncology. So I work with an incredible visionary in my company, Henry Bedford. And Henry's always challenging my vision, challenging me to see bigger, see further. So let's just give me a glimpse of how you see this in the future. I get there's a whole long way to go, but Is this something that you're envisioning can test brand new treatments that will expedite how long it's going to take to get some good data? What are you looking at in the future with this?
Uh, wow, that is a great question, and we have so much in our sights. Something that I haven't really spoken about yet that actually is clinically available right now too is we have an immunotherapy assay as well. So I've described how we're testing drug response against cancer cells. But the thing about immunotherapies, the checkpoint inhibitors that so many patients are getting today, um, the thing about the immunotherapies is that they work to activate the immune system. So they're not really working directly on the cancer cells. They're working on the body's immune system to strengthen the immune system so that it will kill the cancer cells. And so we have sort of another version of our test that when we get that specimen from the sample, we can't do this from tissue, but we can do it from those ascites or pleural effusions, those fluid specimens. Those specimens often contain a lot of immune cells. And so, we pull the immune cells, the T cells, they're called, and we're actually measuring the mass response of the T cells against the checkpoint inhibitors, the immunotherapy drugs. I think that there is an amazing opportunity that exists there that is sort of a short-term opportunity for the company even because those drugs have been phenomenally effective, but they're very hit or miss from patient to patient and they're not always easy drugs to tolerate. But there are a lot of them out there, and the general consensus right now is that all the immunotherapies are kind of the same. But what we're seeing in our early data with this is that we might see one patient that we test four different immunotherapies in, and one of those works and the other three don't. And that is going against a little bit of the standard belief right now about immunotherapy. But you know, what if you're a patient who has tried immunotherapy and failed, and it turns out you just got the wrong immunotherapy? And there's another one that's worth trying. And I think there's really a huge opportunity there. Beyond that, so continuing to build out what we're doing today, adding more drugs, adding more cancer types, continuing to build out our immunotherapy assay and make all of these assays really clinically available and getting them to the hands of doctors and patients that can use them in the front line. That is our primary focus. There is a huge opportunity that exists with drug development. Drug development trials take years and millions of dollars and most pharmaceutical companies, when they're going through drug development, will have different molecules of a certain drug that they're working on. And in the early phases of that development, they might have discovered a certain mechanism of action or certain approach, and they might have two or three different molecules that they're sort of competing with each other on determining which one of these three molecules has the highest likelihood of creating success. Imagine if they could send those three molecules to us, we could test them against samples of live cancer cells from 100 different patients, and 3 days later, um, go back to this pharmaceutical company that's developing these new drugs and say, hey, this molecule hit 85% of the time, but these other two molecules, hit less than 20% of the time. It could really help to streamline drug development. There's a great deal of opportunity that exists there, and we're already having conversations like that with different companies and organizations. And I think there's a huge opportunity that exists in that space. And then the big picture for me is is really facing patients and advocacy. I've been in this space for a long time. I've been in the sort of cancer diagnostic environment for, you know, over 15 years, longer than I'd like to say. Um, and I've seen a lot of really exciting advancements come, but this is probably the most exciting tool I've ever come across. And I think the opportunity to impact patients in the late stage setting is exciting. Buying people a little more time, helping to select those therapies better. Like most people in the world today, have had, close touches with cancer in my family, and to have had a tool like this to help those people out, even if it just bought us a couple of extra months, would have made all the difference in the world. But I have a bigger vision that years down the road of not just using this in the late stage environment, of moving this earlier and earlier in the treatment paradigm, and somebody's newly diagnosed, they're getting a biopsy for their diagnosis, and being able to really personalize care, to not only do you have these these targets that we have target inhibitor drugs for, and you have these genetic mutations, and you have this, and we're going to treat you with this first line treatment, um, but to take that just then a step further and say this drug worked in your cells and we know that it worked, um, could potentially improve response in even early stage disease, which we know improves long-term outcomes. If we can get better responses sooner, it creates a better situation for everybody down the road, and that's where we'd love eventually to be.
Well, the longer we're subjected to a treatment that's not working, the more side effects, and it's sort of a downward spiral. So I love the idea of being able, I love your vision, my friend. I love it. Let's treat it as early as possible. Let's customize it. Let's know what's going to work, and let's be able to test more drugs faster through the get them through the research phase. And I absolutely am excited, hopeful. There's a little bright spot in the future. I know you have a little bit of familiarity personally with somebody in your family with ovarian cancer specifically. You know that we don't get to hear a whole lot of excitement in the world of ovarian cancer. So, this specifically, you've tested for ovarian cancer patients, and it's looking kind of optimistic.
Yeah, we've done a lot in ovarian, and we've seen some really nice responses so far. So it's an area, like I said, we're able to do virtually any kind of cancer type, and we're willing to take just about anything. But as an early company doing a lot of research and ongoing development, um, unfortunately, we live in a world of limited bandwidth and limited resources and limited everything else. And so it is important for us at this stage to really build up our data in a more of a limited group of cancer types. Um, we have every reason to believe that cancer is cancer, in the fact that when I'm testing a real drug against a real cell, I don't care where the cell came from. But for the clinical world, you really do need data that says, here's 200 ovarian cancer patients, and here's the effectiveness of the test in that specific environment. And we're working towards building our data out in that way. Non-small cell lung cancer, ovarian cancer, pancreatic cancer, gastric, and colon. Those are the areas that we're probably most specifically focused on right now. And the reason is that these are all areas that, unfortunately, there's a lot of people out there that have these diseases. And they're areas that just don't have as many options available to them as a lot of other cancer types do. And ovarian is one that is, as you said, close to my heart, and it's a really tough disease. And you get into that later stage setting, I mean, average stage of diagnosis is 3C. And especially for those patients that are not platinum resistant, the opportunities for ongoing drug therapies and just available options become more and more limited, more and more challenging. And so if we can move the drugs that are going to work earlier in that paradigm and get them to those patients sooner and improve those outcomes, extend that life, and that response in any way, then it's really what drives the team, and it's what we're all here to do. Um, we could build a whole company in this drug development world that I talked about a minute ago, just working with the pharmaceutical companies and doing drug development. There are plenty of companies in this ex vivo drug space that have gone down that path. That's not our goal as an organization. We want to continue to be really patient focused, and we want to continue to offer a clinical value that's gonna change things for patients and their families.
I love it. So let's get the treatments that are going to be effective and skip them to the front of the line, get them in the body, get them working sooner rather than later. And I am going to link all of your contact information in the description so that people can reach out to you, because I think this is an exciting time. And I realize it's still very early, but you need data and we need answers. So let's get together on that. And for those of you that are listening, I look forward to our next show with our next guest. So please tune in and check out all the information below so that you can reach out to Dennis Watson. Dennis, thank you so much for joining the show.
Thank you, Melanie. It was an absolute pleasure. And if anybody does reach out, please let me know that you heard about this through Melanie's show and the Overy Jones show. And we'll be more than happy to help answer your questions or guide you through the process if it makes sense for you. But I have an awful lot of people that reach out just to ask questions, and that's perfectly fine as well.
Well, you're an amazing resource and a wealth of information. So thank you so very much. And to all of you, I look forward to seeing you again on our next show.